This video is going to focus on how the three players of translation are going to interact. So remember, with translation, we're starting with a molecule of messenger RNA. And then we are going to divide that RNA into sets of three called a codon. And each codon codes for a specific amino acid that needs to get brought to the ribosome, which you're going to see in just a second. But remember, I told you that when the messenger RNA says, hey, I need a methionine, which is always going to be our first amino acid, we need a transfer RNA to go out into the cell and grab that methionine, and then it will bring it eventually to the ribosome. So how does the transfer RNA know which amino acid it's going to have and where it's going to, when it's time to bring it? Well, there are two important structures or parts to a transfer RNA. The first is up here at the top, the amino acid attachment site. That's where its specific amino acid is going to be attached. So this one is going to be holding on to methionine, and that's all that it's going to be looking for. It's going to go out into the cell, grab a methionine, and wait until it needs to be delivered to the ribosome. So how does it know when it's time to bring methionine to the ribosome? Well, there's a second region of the tRNA molecule responsible for this, and that's the anticodon, which you see down here at the bottom. So notice it also has codon in the name. Whenever you see codon, you need to think 3 nucleotides. If we want to be more specific, 3 RNA nucleotides, because notice it has uracil as well. So what does anti mean? Well, we've heard that before when we talk about anti-parallel. Remember, that meant opposite parallel directions. So anticodon, it means opposite of the codon. So remember, codons are specifically on the messenger RNA, like you see down here. The anticodon is going to be a set of 3 RNA nucleotides that are opposite to that codon. Remember, opposite is just going to mean, in this case, that they base pair together. So check this out. So look at where my mouse is. Do you see how the codon here is AUG? Well, think about base pairing. A always pairs with U when we're talking about RNA, right? Because there's no thymine in RNA. So A pairs with U. U here is going to pair with A, and G is going to pair with C. So if we know the sequence of a codon, we automatically know the sequence of the anticodon on the tRNA that's going to recognize it because of base pairing rules. Okay? So if you think back to my example that we did in the last video, remember we needed methionine and only methionine first. Okay? And then we needed the next amino acid, and I have already forgotten what that was right here. Let's see. Oops, sorry, y'all. Let's go through it real quick. Okay, so we needed proline, and then valine, glutamine, and serine. So this protein would only be correct if we had methionine first, proline second, valine third. Okay, so we have to have that specific sequence. And so that means that we would need, right, the tRNA that's holding on to the amino acid methionine to come in first. And the way that's going to happen is because this tRNA would be holding on to methionine, but it can't drop it off until it encounters an AUG, because that's the only place its little anticodon will fit, because only UAC is going to base pair with AUG. So anytime AUG comes up, this particular tRNA, which would be carrying methionine, is able to bind. Because if you look at this next one, okay, and I'm the example on the previous slide that I just showed y'all was created independent of looking at this slide. It's just AUG always comes first. So in this example on this slide, the second codon is CUG. Do you see that here? Look where my mouse is, CUG. So the only tRNA that can fit here is going to be the one that has the anticodon GAC, because it's going to be opposite or base pair with this codon. And then it will bring whatever that amino acid is carrying to the ribosome. Okay, and we're gonna do a visual of this again, I believe, in the next video. All right, so we've got our two parts to the transfer RNA. Again, I'll remind you of these later and how they interact. Once the tRNA has the amino acid and it knows when it's its turn, it's going to bring it to the ribosome. Okay, so notice here's the ribosome. There are 3 little binding sites on the ribosome. Well, there's 3 pockets on the ribosome. There's 2 actual places where a transfer RNA molecule can bind. That's in the P site and the A site. So remember, we talked about the ribosome being made up of 2 pieces. So here's the small ribosomal subunit. Here's the large ribosomal subunit. Notice the messenger RNA is here, associated with that small subunit. And then we've got transfer RNAs hanging out in the ribosome, bringing their little amino acids, which are the little purple shapes you see here. So notice we have all 3 of our key players interacting so that the process of translation can occur. So let's go ahead and look at this process a little bit more detailed. There are 3 steps to translation: initiation, elongation, and termination. Turns out these were the same 3 steps, well, the names at least were the same, for transcription. So when we think of initiation, the purpose is to initiate the process. That means we have to bring all 3 key players together. So in order to do that, we're first going to have the small ribosomal subunit and the messenger RNA are going to bind. Once that happens, you'll see the start codon over here. Excuse me. And once the start codon is there, that's going to allow the first tRNA, which we call the initiator transfer RNA, to come in and bind. Okay, because look at the picture closely, y'all. Do you see where this AUG start codon is? Notice it lines up with this big binding site, the P site, in the ribosome. So once the AUG is there, because the messenger RNA and small subunit have gotten together, that's going to allow that initiator tRNA to orient itself just right so that it's snugly in place. Okay, but the large ribosome hasn't actually binded yet. Okay, the initiator tRNA actually gets there first. Okay, but it's just kind of lined up for where it needs to be. Okay, so again, first messenger RNA and the small ribosomal subunit bind. This is going to expose that start codon. So next, the initiator tRNA comes in and it's going to interact with that start codon because the sequences are complementary. And it's just going to kind of hang out there. Once that happens, now the large subunit can come in and bind, and it's going to bind so that it's attached to the small subunit, but also that hanging tRNA that's there, it's going to orient itself so it's in the P site of the large ribosomal subunit. Okay, so the large subunit comes in last and it orients itself so it's in the right place for the small subunit and the initiator tRNA. Once all three of those are together, then we can move on to the next step because we now have all three key players hanging out together. So notice, take a close look at the picture. Okay, there's lots going on here. We have our initiator tRNA carrying that methionine, because notice the tRNA is associated with the start codon, and it has methionine, which will always be our first amino acid. In this example, the second codon is CUA, because that's on the messenger RNA. So we know if that sequence is CUA, then the transfer RNA, its anticodon, will have to be complementary to that, which is GAU. Okay. Turns out this transfer RNA is going to be holding on to leucine. That means that leucine needs to be brought to the ribosome next, which it has been, and methionine is going to get connected to that leucine. Once that connection has been made, that connection is called a peptide bond. Once that's been made, now this initiator tRNA is empty. Notice there's no amino acids attached to it. So it's going to be kicked out of the ribosome. If we go to the next slide, it just shows that same picture at the top. So that empty tRNA is going to be kicked out of the ribosome. That's going to allow the current transfer RNA that's hanging on to both amino acids to scooch over so that the third tRNA can come in and bring that third amino acid with it. Okay? So notice how these three key players are constantly interacting. Okay? If you look at this little arrow down here, notice the messenger RNA is kind of being fed through the ribosome so that once the amino acid's been dropped off, we need to make room for the rest of the amino acids to come through. So that's why you see this AUG down on the bottom. See how it's moved out of the ribosome, because it's making space for the rest of the codons to be read. Okay? So just remember, the messenger RNA has the codons. This is what you would look at and use that codon wheel for to figure out which amino acid is next. That amino acid gets delivered by a transfer RNA that's holding on to its respective amino acid, but it actually drops it off at the right time because the transfer RNA has an anticodon, which is opposite of the codon on the messenger RNA. So messenger RNAs have codons, transfer RNAs have anticodons. All right, so this same process is going to keep happening until it's reached the end of the RNA molecule. And remember, that final codon is called the stop codon. So check out this picture. Notice all of these amino acids have been added, and at this point, it's done. We've made the protein. So that means we need to tell this process to stop. So there are 3 different codons that code for a stop codon. But remember, there's no amino acid called stop. So notice when this stop codon is in the proper position at the ribosome, notice there's no tRNA that's coming in, because tRNAs only carry amino acids, and there's no amino acid called stop. So something else called a release factor, I would write that down. Something else called a release factor will come in here and bind, and that is going to trigger the ribosome to fall apart, or more like disassemble, and that's also going to allow the little chain of amino acids down here to also come apart. Okay, so the chain of amino acids stays intact, but it releases from that final tRNA molecule. So kind of think of the release factor as like cops, right? So if cops arrive to the party, in this case, the party of translation, it's like, whoa, 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 sorry, party's over. We'll turn the music down. Everybody's going to go home. Okay. So when the cops arrive, again, the release factor arrives to the ribosome, everything's going to go its separate way. The ribosome is going to disassemble to the small subunit and the large subunit. The tRNA is going to release the chain of amino acids. The tRNA is going to let go of the ribosome as well. All of the key players go their own separate way, and the process is done. All right, y'all. Let me know if you have any questions. Bye, y'all.